The overall goal of this procedure is to produce and purify BDNF habitat in an optimized protocol involving transfection of HEC293 cells and purification in a chromatography column. The BDNF habitat is then mono-Biotinylated by an in vitro reaction for posterior labeling and detection by fluorescence microscopy.
The main advantage of this procedure is that it allows the production, purification, and mono-biotinylation of BDNF in an optimized manner. The transfection parameters have been optimized for maximum BDNF production using a cost-effective transfection reagent, and the purification process is performed in a chromatography setup that facilitates manipulation and allows to escalate the procedure.
Begin by diluting 60 micrograms of polyethylenimine in 500 microliters of unsupplemented DMEM and 20 micrograms of BDNF habitat plasmid in 500 microliters of unsupplemented DMEM per 15 cm dish to be transfected. Incubate for 5 minutes at room temperature and then carefully pipette the DNA solution into the polyethylenimine tube, mixing once by up-down motion. Incubate for 25 minutes at room temperature and then drip 1 milliliter of the pDNA mixture throughout each 15 cm dish. Incubate the cells with the pDNA mixture for 3 hours and then change the medium to incubation buffer. 48 hours after the transfection of the cells, collect the medium from all the dishes.